So welcome back to our next lecture. The last time we met, we were looking at features of nucleosides, features of purines and pyrimidines. Last time we were looking at purines and pyrimidines being hydrogen donors and hydrogen acceptors. The last property that we can look at now is the fact that when you look at these pyrimidines and purines, they are all hydrophobic. Therefore, they can interact with one another in a feature that is referred to as hydrophobic interaction. Right, now let's proceed and look at nucleotides. Nucleotides. So, what are nucleotides? By definition, we can say that these are phosphate. Okay, these are phosphate salts of nucleosides. Nucleoside. So a nucleotide is just a salt of a nucleoside. Okay. For instance, if you add a phosphate to a nucleoside, you form a nucleotide. Okay. Example, if we look at cytidine, okay, I'm writing cytidine, or is it I'm drawing cytidine? This is cytidine. Okay, that is cytosine connected to a ribose sugar. This is our ribose sugar. Of course, that is your terminal CH2 hydroxide. Now, this can react with the ATP. What the ATP is doing is just to transfer one of the phosphate to the terminal hydroxide, and the ATP will be converted to ADP. Of course, you require an energy before you require an enzyme, because this is an enzymatic reaction. Okay, since we are using cytidine, okay, the enzyme we require is a cytidine kinase. Cytidine kinase. So the product, which is a phosphorylated cytidine, is formed. So this is our phosphorylated cytidine. Phosphorylated cytidine. Okay, so our cytidine is now attached to a phosphate group. This is our phosphate group. Okay. Right. Okay. So cytidine has been converted to a nucleotide. A nucleoside has been converted to a nucleotide. Okay. This nucleotide So this nucleotide, we can call it as it's coming from cytidine. Okay. So what we need to note is that what have we done to this cytidine? We have just phosphorylated it. Phosphorylation. The reaction is a phosphorylation reaction. Where have we phosphorylated this? We have phosphorylated it at 5 prime. Remember, this is 1 prime, this is 2 prime, this is 3 prime, this is 4 prime, this is 5 prime. So at 5 prime, that's where we have phosphorylated the cytidine. So would then call this as cytidine. Okay, we have phosphorylated at 5 prime. How many phosphate have we added? 1. So cytidine 5 prime monophosphate. Monophosphate, which you can say CMP, side D monophosphate. So this is a nucleotide. So in a nucleotide, what bonds do we see? We see this bond. This is a beta N glycoside or glycosidic bond. Okay, right. Then we also see this bond here. This is an ester bond. Okay. Okay. So two bonds that we see. Okay. Right. Let's look at another one. Or let me just say the rest will occur in this manner. The rest will occur in this manner. Okay. Let me give also an example from there. This is a pyrimidine. Let me give an example from a purine. Okay. An example from a purine. I'll use the example of adenine. Okay. This is my adenine. Adenine. This is my adenine. Okay. That's my adenine. The adenine is connected to a sugar to form adenosine. Remember, time we talked about nomenclature. Okay, so you have the terminal CH2 hydroxide. Okay, so that is the adenosine. Adenosine. This can be phosphorylated. Okay, ATP will transfer one of its phosphate to form ADP. Of course, we will require adenosine kinase. Adenosine kinase. Okay, just like we saw with the purine, the pyrimidines. Similarly, with the purines, there is going to be phosphorylation. Phosphorylation takes place, okay, at the 5' carbon. 5' carbon. Okay, this is my 5' carbon. So I add the phosphate here. One thing you observe is that, look at the way I've written these oxygens. I told you when I was talking about phosphoric acid, it dissociates, okay, to form dihydrogen phosphate, then do hydrogen phosphate, and then to phosphate. I told you that at pH of 7.2, this is how it is occurring. So in the body to have this kind of scenario. Okay, so what we have formed is adenosine at 5'. 5' is here. Is 5' and 4' end. 4' carbon, 3' carbon, 2' carbon, 1' carbon. Here we have added how many phosphate? Only one, so we call it monophosphate. So this is another nucleotide. This is another nucleotide. But it's possible also that you can carry out another phosphorylation. So ATP changes to ADP. Remember, I said initially that this is a phosphorylation reaction. We would say this is the first phosphorylation reaction that has occurred. Okay. Similarly, if we added another phosphate group, okay, the phosphate will be added here. Okay, so I'm bringing the structure here. Okay, so now we will adenine there. Adenine there. Okay. Adenine there. Okay. CH2. So we have added another phosphate. Okay, so that is a structure that you have formed. Okay, that is a structure that you have formed. 2 phosphates have been added. So then we would say this is the adenosine 5 prime. At 5 prime, you have added 2 phosphates. So diphosphate. Diphosphate. So we can short it as or abbreviate it as ADP. This is AMP. Okay. Right. We can also phosphorylate this structure. We can phosphorylate this structure. Again, ATP converted to ADP. It's an enzymatic reaction, and then we produce another nucleotide. So we have now added, if we use this, if we do this phosphorylation, the third phosphorylation reaction, then we are going to have 3 phosphate groups added. 3 phosphate groups added. Okay, 3 phosphate groups added. So we'd call this as the nucleoside is adenosine. Okay, then at 5 prime, we have added 3 phosphates, triphosphate. And this is what you call commonly known as ATP, the universal carrier of energy. We know this from grade 9, or is it from grade 1 nowadays? Okay, that is your ATP. Okay, so this is a nucleotide. This nucleotide is a triphosphate. This nucleotide is a diphosphate. We had another one which was a monophosphate. Okay, so the bonds that you see here, you see a bond here, we call this as a acid anhydride bond. But we see another one here. It's also an acid anhydride bond. Okay, but we also see this bond which I mentioned earlier. It is an ester bond. Then we also have this, the beta N glycosidic bond. This one here. So you see 4 bonds. Okay, beta N glycosidic bond, ester bond, acid anhydride bond, and acid anhydride bond. In a diphosphate, you only see 1 acid anhydride bond. In a mono, you don't see the acid anhydride bond. So have that in mind. Now, in order to distinguish which of these phosphates, for instance, we want to talk about, we need to name it. So the phosphate next to the terminal carbon is called alpha. The next one is called beta. The third one is called gamma. So we have the alpha phosphate, the beta phosphate, and the gamma phosphate in the triphosphate. So these are nucleotides, which are basically the phosphate sorts of nucleotides. Or we can say the sorts of nucleosides, not nucleotide, the sorts of nucleoside. Okay, so we have distinguished between a nucleoside and a nucleotide. In a nucleoside, you have a nitrogenous base and a sugar. Okay, in a nucleotide, you have a nitrogenous base, a sugar, and a phosphate. The phosphate, you may have one, you may have two, or you may have three, as it were. Okay, right. Maybe let's now look at the uses. Let's look at the uses of nucleosides. Okay, the uses of nucleosides. Or let us look at analogues. Analogues of nucleotides. Okay. What comes to mind? What can I think of? Okay. So, firstly, I'm giving you the normal nucleotide. Or rather, the ordinary, not the normal. There is no abnormal. The ordinary nucleotide. Okay. We said this is it. Adenosine 5' di. Sorry, there's only one phosphate, so we'll call it monophosphate. Okay. So a structure was designed that is similar to this. Okay, a structure similar to this. So let's look at the structure similar to this. Okay. Okay. So you have this bond. Okay. Are you with me? Then you have this bond where there is oxygen. So you replace this oxygen with carbon. Are we together? You replace this oxygen with carbon. So there is carbon there. I do like that. There is carbon there. Okay. And then after having done that, I delete everything here. So, and then introduce this group. I introduce a phosphonate. This is called the phosphonate. Okay, this is a phosphate. This is a phosphate. Are we together? This is a phosphate. Okay, this is a phosphate. Let me draw it in the way that I've drawn it there so that you can see. So I have this phosphate, double bond, that. This is phosphoric acid where the phosphate comes from. Okay? But the phosphonate, phosphorus is joined to carbon, and then the phosphate there. This is the phosphonate. Okay? That is the phosphoric acid, the area one. This is phosphonate. So I introduce the phosphonate there. This is the phosphonate. Okay? Right, so I introduce the phosphonate and remove the rest. So I form what is known as the adenovia. Okay, adenovia, which is an analog nucleotide. That is an analog of a nucleotide. Okay? Does it have the three components? Does it have a base? It has adenine. Then does this component, which sort of is a sugar, you can say this is a sugar that has been modified. And then it has the phosphonate. Okay, it has a phosphonate, which is similar to phosphoric acid. So it is an analog of nucleotide, which was used for treatment of cancer. Another one that was devised is it's very similar to the first one. Very similar indeed. Okay, so we have that. Sorry, let me try to take it a bit downwards. I have this, and the phosphonate there. Then here I introduce the methyl group. Okay, the methyl group. So this is known as tenofovia, a very important analog of a nucleotide which is used in the treatment of HIV. Okay, this is a nucleotide analog. The other ones we looked at were nucleoside analogs. Okay. Enough about the nucleotide analogs. Okay. So we looked at the nucleotides. Okay. We looked at the nucleotides. These nucleotides can react with one another. Remember where we are coming from. From the introduction, I said that nucleic acids, let me say it again. I said that nucleic acids are polymers. Nucleic acids are polymers of nucleotides. So at this point in time, we have looked at the monomers. Okay, these are monomers. The monomers of nucleic acid, which are the nucleotides. What we've just finished looking at are monomers of nucleic acids. Okay? So the use of these nucleotides is to produce nucleic acids. So that they can be added together to form nucleic acids. So let's look at how nucleic acids are formed then. How the polymerization occurs. How does the polymerization occur? But before I go to that aspect, let me look at the uses of nucleotides. The uses of nucleotides. So we have just said that nucleotides are used to synthesize nucleic acids. So nucleotides are monomers. Sorry, they are monomers. They are monomers of nucleic acids. So we use nucleotides to produce nucleic acids. I'll look at how this occurs just after this. Secondly, we can say that nucleotides are structural components. Okay, nucleotides are structural components. That's a use. Structural components of a lot of coenzymes. Maybe at this point, let me give you a few examples of coenzymes. Okay, a few examples of coenzymes. Okay, we say that substances such as NAD, the coenzyme. Okay, Fu is nicotinamide. Adenine, dinucleotide, which is a coenzyme. The other coenzyme that we know is NADPH. NADPH, in short, that is nicotinamide, adenine, dinucleotide, phosphate. Okay. The other one that we know is FAD. Okay, which is flavine, adenine, dinucleotide. Other ones that we know include the coenzyme A. These are but a few examples of coenzymes. Okay, these are examples of coenzymes. NAD is a coenzyme, NADPH is a coenzyme, FAD is a coenzyme, coenzyme A is a coenzyme. Okay, coenzyme A is made up of 3 components: cysteamine, beta alanine, pantothenic acid, and of course we have adenosine also there. Okay, we have adenosine there. Right.